my congratulations on passing through the first third of our course. So we had, I'd say, quite a complicated first third that was devoted mostly to to some chemistry, to organic chemistry, and we obtained some basis of the organic chemistry. And now I guess it's time to get a little bit closer to the biochemistry. And we start with this quite an interesting example. This is a map of known, I guess not maybe all known, but some of the known biochemical pathways that are present in the human body and in our cells. So you see there's a lot of different things going on here, and of course nobody knows exactly what happens in each of these exact pathways, so probably you can find some articles that describe some parts of these pathways and so on, but of course we don't expect to learn them by heart or something like that. What's the purpose of today's lecture? So first of all, I want to talk with you about the metabolism. That's pretty good and that's pretty biological. But there is the second reason. I want to show you how deep can we get inside of some of these pathways, and and what can we understand if we study, for example, some articles on that pathways and so on. And these few pathways that we look today, that's glycolysis, that's Krebs cycle. I guess we'll get to Krebs cycle tomorrow, but I hope that we will be able to get through glycolysis today. And they will be exemplary to show you what can we learn and what can we know about what happens in in our cells and how all that things work. Okay, so with that being said, let's get to metabolism. So first of all, food fuels, they are degraded and large molecules are constructed step by step in a series of linked reactions, and they are called metabolic pathways. So basically all you saw on that map, that were metabolic pathways, so how one molecules are transformed in another molecules. An energy currency common to all life forms, at least all that we know, is adenosine triphosphate, ATP. And we will see these kind of molecules a lot. I will talk today about glycolysis and next time about Krebs cycle and phosphorylative oxidation, and basically that will be a way when oxidation of carbon fuels will power the formation of ATP. We can do that without oxygen, we will see that today, but of course we prefer to use oxygen because that gives us much more energy. That's much more energetically favorable. And also there are many metabolic pathways, a limited number of type of reactions and particular intermediates are common to many pathways. So I will show you today some sort of examples, and you will see that many patterns, they are repeated even throughout one pathway. And of course let's put it this way, if evolution finds some way to do something, it will reuse that way many times more, so we will see that more and more. And the last one, the pathways are highly regulated, and I guess we will see that as well. Here are a few links to atlases. You can look, you can use these links to get to some sources that show different pathways that we know, and probably you may want to explore some of them. Okay, while it's loading, let's go further because I have a really large lecture today. Okay, so living organisms, of course, require energy because we are living, and we need that to perform, let's say, a mechanical work, so like muscle contraction or like movement. Even if we don't move, we need energy for active transport of molecules and ions. For example, right now I'm thinking, and a lot of different ion channels are opening, closing, pumping ions all over around my brain, so I need energy for that. And of course, synthesis of macromolecules and other biomolecules from simple precursors. So we all are changing, growing somehow, so on and so forth. So we need somehow to update the chemical composition of our organism. We need to recycle some molecules. We need to create some new molecules and put them somewhere. So that's basically all that needs some sort of energy. Those reactions that transform fuels into solar energy are called catabolic reactions, or more generally catabolic. So here is a scheme. So you have some fuel that may be carbohydrates. We will talk about that today. That may be fats. Maybe we will talk about that later. And during catabolism, you get CO2, water, and some useful energy. The reactions that require energy, such as synthesis of glucose, we may need that as well, or fats or DNA, they are called anabolic reactions or anabolism. So we have some useful energy, we have simple precursors, and we construct some more complex molecules. Okay, what's the problem with constructing new molecules? They require some energy. But now let's think for a moment, how do we invest energy into creating some more complex molecules? In organic chemistry, we could, I don't know, make some higher temperature or use some pressure or whatever, but we cannot do that in biological system. So I cannot just heat myself to 100 degrees C to make some reactions to go. It's not an option. So what do we do if reaction requires energy? And there is an interesting twist. It turns out, oh, I see some comment. Enzymes. Yes, enzymes are the key here, but please remember, enzymes do not shift the equilibrium. So wherever equilibrium is, there it is. Enzyme does not shift it. It just helps to acquire that equilibrium. So what is actually happening? Enzymes are able to couple series of reactions so that you have one reaction that requires energy. For example, this one, you need to use some 5 calories per mole, and some different reaction that is energetically favorable. And when these reactions are coupled together, so here is an example of this scheme, if you couple them together, you can get a new reaction that is now energetically favorable. So in the expense of that energetical favor of one of the reactions, so this one, B to D, would really like to go from B to D because that's really energetically favorable. So B is here, D is somewhere down there, while this one, the first one, is not. So A is somewhere and the products are somewhere here, B plus C. But if we couple these reactions to make from A C plus D, we will be able to get our final state somewhere here. And now this is energetically favorable. So that's the key point. So many reactions, they go this way. So we couple the reaction that we are able to perform because it's energetically favorable and a reaction that is not favorable. And here is an example of, let's say, a universal reaction that we are trying to couple to many other reactions to get them going. This is the reaction of the hydrolysis of the ATP. That's the reason why I told you that ATP is some sort of universal energy currency. So you use ATP, you hydrolyze ATP. These hydrolysis, you see there is some energy like minus 30 kilojoule per mole. Or you can hydrolyze that to AMP, but that's a rare case, so mostly to ADP. Here are the chemical formulae on the left. So here you see ATP, And you can see the adenine in blue, ribose in black, so with sort of sugar, and you see that different number of monophosphate units gives you triphosphate, diphosphate, or monophosphate. Some biosynthetic reactions are driven by GTP, UTP, CTP, but they are pretty the same in terms of energy. So you should not bother with that. Good. The high phosphoryl transfer potential of ADP, so 30 kilojoule per mole, that's pretty okay, is explained by resonance stabilization of orthophosphate. So I hope you remember from our first third of the course what is resonance stabilization. And I guess I showed you even the orthophosphate example, probably. Electrostatic repulsion. So they are all charged. So if you break the bond and they are able to get away one from another, that's probably energetically favorable because you don't have that summand that comes from Coulomb interaction. Increase in entropy. Don't underestimate this one. So you had one molecule, now you have one molecule and one inorganic phosphate, we call it. So you have now two molecules, and that basically brings you an increase in entropy because you have a higher number of microstates in your system. And I saw some article, I don't have it at hand, but there were tables that were comparing different where from comes the Gibbs free energy, and you will see that entropy makes, let's say, large investment in that. And of course, stabilization due to hydration. Interesting thing that ATP is often called a high energy phosphate compound. Nonetheless, there is nothing special about the bonds themselves. They are high energy bonds in a sense that much free energy is released when they are hydrolyzed. Here are two schemes that show you where is ATP on scales of energy. So here are a few other molecules that may be used, for example, for storing some energy. So here you see ATP, here you see phosphoenol pyruvate, we will see that today, and some other molecules. And here is the table that compares these energies. You see that PEP, or phosphoenol pyruvate, has like minus 61 kilojoule per mole energy of hydrolysis, while ADP has minus 30. That's ATP to ADP. Interesting question for you. How do you think, why do we use ATP, for example, but not phosphoenol pyruvate? It is more energetical. There's more energy stored in that molecule. Why don't we use that as a universal energy currency, but mostly most processes stick to ATP? What may be the reason? No. Not. Yes, adenosine is used, but it's not the reason. Okay, I'll give you a hint. How do you charge your phone? So you probably use a charger, yeah? So you don't plug it directly into the socket. So the reason is that other molecules like phosphoenol pyruvate, they are too energetic. So you see, you're coupling two reactions, and what you want to get at the result, of course, the reaction of the hydrolysis should release more energy than you will spend on the coupled reaction. That's the first point. The second point, you don't want to release too much energy because you will just lose it. So yes, you'll be able to perform second reaction, but the rest of the energy that you have not used will be just, I don't know, transformed into heat. So it just dissipates. So ATP is has a really nice balance. So this energy is already enough to power most useful reactions. So it is large enough already to do that, but it is lower from the other chemicals, so you don't dissipate too much energy. So that's the reason. You use charger to charge your phone. You need 12 volts to charge your phone. You don't want to charge it from 220. You will, first of all, it will just break down. And if you even manage somehow to do that, you will probably lose a lot of energy into nothingness. So that's the reason. Okay, and a really nice take about the adenosine, but from a little bit another perspective. You see, if you have ever read some, I don't know, some books or articles on the abiogenesis, so how some living things can come from some non-living things, you should know about the theory of the RNA world. So that sometime ago, very, very long time ago, we existed in the form of some sort of RNA molecules. And this idea, I guess, some support to that idea may be that activated carriers such as ADP, NADH, FADH, and coenzyme A contain adenosine diphosphate units, probably because these molecules evolved from the early RNA catalysts. So non-RNA units may have been recruited to serve as efficient carriers of activated electrons and chemical units, a function not readily performed by RNA itself. And we can picture the adenosine ring of FADH2, for example, binding to uracil unit in a niche of RNA enzyme or ribozyme. I guess I told you something about ribozymes in previous lectures, so probably that part was to fit into some sort of RNA structures, and the rest was basically performing that energy carrying duty. Here is one more example of what do we use when we perform some exercise. So in the initial seconds of the exercise that we do, we are basically using ADP to power our muscles. Then it drains, and there are certain reactions that use creatine phosphates to regenerate ATP. So I don't think this plot is really okay, because it shows that ATP goes to 0. It does not. It is regenerated from creatine phosphate, so there should be something a bit different drawn here, but anyway. And indeed, the creatine phosphate is the major source of phosphoryl groups for ADP regeneration for a runner during the first 4 seconds of 100 meters sprint. And that's basically comes all down to these sort of equations. Good. What else? Most interchanges of activated groups in metabolism are accomplished by a small set of carriers. Here are some examples of carriers, and you see that they have in many cases they have a vitamin precursor. So when you are taking some vitamins, you can think that basically some sort of group carriers will be synthesized from that. Here is just to show you some table with vitamins and that are working as some sort of coenzymes in certain reactions, and type of the reactions that they are involved in. And here are some consequences of the deficiency, like beriberi, so on, so forth. So many different bad things happen when you don't have enough of some of these vitamins. So here are some more, and you see that different functions of these vitamins. So I'm not going into the detail because it's not the topic of this lecture, but probably after the lecture you would like to look these tables. Vitamin C is quite tricky. Basically, we have almost all the needed pathway to synthesize vitamin C on our own, except the last step. So the last step, enzyme, is broken. It's not a problem because we get a lot of it from different fruits and so on, so it doesn't trouble us in normal conditions. But it turned out that people can find such a conditions that these becomes a problem. So you know that sailors had troubles with scurvy, and in far north, if you read, I don't know, some books of Jack London, you probably will get to the point when they have some scurvy or some troubles with scurvy. So that's basically the deficiency in the C vitamin. And what's interesting that why do I say it's tricky? Because it's really hard to reproduce in animal models. We have broken enzymes, and probably, I don't know, capybaras have this enzyme broken, and one more animal, I don't know, I don't actually remember, so it's not so many animals that you can reproduce the problem and study that. So yeah. Good. What else? We will consider, I hope we will consider today aerobic oxidation and anaerobic oxidation. You might have been thinking that anaerobic oxidation is something exclusive to some bacteria and archaea, and today I will show you it's not. We are doing that as well, especially during some sort of exercises. And yeast, they can also do aerobic or anaerobic oxidation. When oxygen is unavailable, yeast and some other fungi can switch to anaerobic growth and produce ethanol as a byproduct. So if you have ever done on your own, I don't know, some wine or whatever, some moonshine, so probably you used this sort of ability, and you have covered the the volume where you keep all that yeast and everything inside, just not to let oxygen to get into there and to do aerobic oxidation. So you should force them to do all the processes anaerobically. Good. Here is the general scheme of what are we doing. So hopefully today we will get through this part. This is called glycolysis. So all these part, that's glycolysis. You may see it's some sort of split into two subparts. We'll get to that. That's really true. And I hope that tomorrow we'll be able to get through Krebs cycle, and you will see how chemical reactions can run in a cycle, what's interesting. And we will get through this sort of electron transport chain, ATC, and we will talk a little bit about processes when you can, I don't know, play with an electron, just throwing it from one molecule to another, and that basically does some pretty decent job, like pumping out the protons and so on. So we'll get to that. Good. Now, glycolysis. It's a pretty ancient mechanism of getting energy, and you can see that because it's highly conserved in different species. The first point, clinical importance. So glucose is the preferred source of energy for most of the body tissues. Brain cells derive the energy mainly from glucose. They can do a little bit something from so-called ketone bodies. I will not get into that, just know that there is this sort of workaround, but mainly from glucose. And the main organ that takes, I guess, most of the glucose from your bloodstream will be the brain. And of course, when glucose metabolism is deranged, it leads to some sort of life-threatening conditions. So definitely, nobody would like that. And normal fasting plasma glucose level is something like 70 to 110 milligram per deciliter, and you will probably see these sort of numbers if you ever do some sort of analysis anywhere. So biochemical analysis in any clinic. And after heavy carbohydrate meal, it may rise, but in a normal person, this level is always below 150. If it gets higher, that's really a problem. So it's a problem because glucose may start different let's say bad reactions, glucosylating some sort of proteins that you don't want to be glycosylated, and doing some other nasty things. So it's really good when it's everything in the bounds that it should be. Good. Regarding glycolysis. So it's performed in the cytoplasm of, I guess, all the cells that we have. But what's interesting, first of all, glucose should somehow get into them. So here is an example. Let's say everything starts with the glucose uptake in small intestine. So we are eating something, so everything is, the food is broken, it is somehow degraded into smaller units in the process of digestion. So for example, proteins are hydrolyzed to amino acids, so polysaccharides are hydrolyzed to simple sugars as glucose, and lipids are hydrolyzed to glycerol and fatty acids. And after the degradation, we can absorb these products through the cells of the intestine and distribute through the body. So here is an example of such a cell. You see here a symporter that transports one molecule of glucose and two sodium ions inside the cell. Then the cell pumps out everything into the blood. So it pumps out the glucose through this sort of transporter GLU2, and expels sodium by another pump, sodium potassium ATPase. These all gets into the portal vein that goes to your liver, and then after that it may go into the general bloodstream. So what's interesting, the first pass of everything you have eaten always goes through liver. So if you will ever study some sort of medicine, pharmacology, being exactly, you will hear the term bioavailability, and this bioavailability basically will describe how badly liver, let's say, what does liver to your drug on the first pass. So it brings down some coefficients into your formulae that tells you that when you have eaten some sort of tablet, it passes first time through the liver, and basically maybe part of that drug may be degraded on the first pass. Then the degradation pattern at least changes a bit, and you use some different formulae, but the first pass is taken into account in the so-called bioavailability. Good. Now, glucose is in the bloodstream. Now all the cells want to get some share of that glucose, because everyone wants some part of energy. All cells want to function somehow. They do this through specific transporters. So here is some schematic picture of the transporter that uptakes the glucose from the bloodstream and transports into the cytosol, where basically we will start our glycolysis. These transporters can be pretty different. Here is the table of major classes of these transporters. You see that they are distributed in different tissues, so depending on the type of the transporter you can guess the tissue. They all have different Km. What is Km? Who will remind me? We had a lecture on enzymes. Oh, I see. No. Can't open. In some sense, but there is a basically term. How do we call these KMs? Do you remember that part of lecture, I don't know, lecture 5 or something like that, on the enzymes? Yes, Michaelis constant, absolutely correct. So Km is Michaelis constant. And do you remember what is Michaelis constant? What does it mean to for some protein to have, let's say, enzyme? Okay, so let's return to the previous lecture. For some enzyme to have high Km or to have low Km. Half of the Vmax. Yes, absolutely correct. So this is half of the that's let's say put it another way. So when the concentration of the substrate is equal to the Km, the velocity of the enzyme working is half of the Vmax. So that's, let's say, more precise definition, because this sort of definition tells you what. So if I have high Km, what does it mean? What should be the concentration of the substrate for the enzyme to work quickly? Yes, so concentration should always also be high. If you have low Km, enzyme starts working efficiently at low concentrations. So here you might see that looking at these different tissues, so that, let's say, neurons, they have very low Km. So brain uses almost exclusively glucose, and it's a really tender tissue, so you don't want to lose it. So that's why you have all these transporters with very low Km, and they work at that very low Km, transporting whenever there is even a little bit of glucose in the blood, because other tissues, they can survive if left for some time without glucose, but brain will not. And you definitely do not want to damage that, because probably that's the only our evolutionary advantage that we as humans have. On the other hand, you can find some, I don't know, where's high Km? Here's the one, GLUT2 in liver or pancreatic, in kidneys, small intestine, and so on. So in liver, we have some sort of storage of the glucose. I'm not sure if I will have time to tell you about that, but there is some sort of of storage of glucose, and we want these transporters to work only if we have high concentration of glucose in the blood, which means what? We have just eaten something and we have a lot of glucose. So, of course, all this glucose cannot be used immediately for performing some sort of work, so we can store that for later. So, if we do not eat for some time, we can then use that storage and basically function okay until the next meal. Good. Here are examples of normal role and clinical relevance of these different transporters. So basically what happens when they are broken. I'm not going into that. And here is the biochemical pathway that we will right now consider in details, at least so detailed as we will be able with the time of lecture given. So first, just few words in general. So okay, we have just did what? So we have digested something. We have uptaken the glucose into the bloodstream. Now from the bloodstream it was uptaken with some sort of cells. So whatever it is, almost everything uses glucose as fuel. Now glucose is in the cytoplasm and we are starting our glycolysis. There are two, let's say, parts of this glycolytic pathway. The first part, you can see that you are performing some sort of, let's say, investment. So here we use ADP to perform some work. So we spend energy to perform certain work. So we will get through each of these reactions, and and I will tell you a little bit about them. But you see that we spent in this part, we spent some energy. Then we get to the second part of this pathway, and here you see that we obtain some energy. Now, interesting question for you. If you carefully examine the thing, you see that on the left I have two molecules ATP spent, so transformed into ADP, and on the right I have two molecules ADP transformed into ATP. So how come can it be that this path leads to some energy gain? Can you see the trick? Let me give you a hint. Oh, I see an answer. Good. Stage 2 is doubled. Yes, stage 2 is doubled. That's correct. You see, here is a sugar that contains 6 carbon atoms. It is transformed into these two molecules that have 3 carbon atoms. And each of them then is processed by this second part of the pathway. So basically you get to ATP, but you get 2 ATP for this molecule. Let me clean this, for this molecule, and you get 2 ATP for this molecule. So all in all you get 4 ATPs. So in your investment step, let's call it this way, you spend 2 ATPs, and in your payoff step you get back 4 ATPs. So basically you get 2 ATPs for all the work performed. Good. What else? I don't know. Let's go through steps. Maybe we'll return to this slide time to time so that we can get some idea. Okay, so glucose to glucose 6 phosphate. So performed by hexokinase. The glucose enters cells through specific transport proteins, and they have one principal fate. It is phosphorylated by ATP to form glucose 6 phosphate. Once again, please note, here we have reactions coupled. So the thing that I told you at the beginning of the lecture. So we are coupling reaction of phosphorylating glucose with the reaction of dephosphorylating of the ATP. That's why we get some gain in free energy. But we spent ATP. Okay, what's interesting, this part basically is performed to, let's let's say it's so. So we have glucose somewhere. Oops. Так, у мене відпав zoom. Не понял. Поча інтернет пропав.
Recording in progress.
Okay, I'm sorry. I guess some troubles with internet. I'm not sure what happened. Okay. I hope it's some sort of temporary problem. Okay. Yes, so the general idea is the following. So glucose gets into the cell. Now you want to restrain your glucose from going away from the cell. Because all these transporters, they can transport one direction, another direction, so you don't want it to get all over the place. So the first thing the cell does is basically adds a phosphate group to the glucose, and now it is trapped inside the cell. So the meaning of the first step is basically to block glucose from leaving the cell. So we are now processing this molecule. We need it to be here. It's performed with the hexokinase. Here you see how nicely this molecule looks like. So these are basically two lobes of the hexokinase. I'll show you a small video of how that works. If internet allows us, because probably it will not. Yeah. No, something working. Good. So here you see how the glucose gets inside into the hexokinase, and you see how the molecule, the protein changes its conformation, and then the reaction is performed. Good. And here is the mechanism of the reaction. I'm a little bit afraid of using all these links. I don't know, let's try, but who knows. Here are just some links for you that show some 3D models of the proteins that we will see. Okay, once again it's loading. It's loaded. Good. Okay, so here you see the hexokinase, and these are basically two conformations of the hexokinase when the reaction is performed. So I guess you can look better after the lecture. Oh, and this one I have totally forgotten. This is one of the atlases of the pathways that I gave links to you at the beginning of the lecture. So it happened that it loaded. and here you see some energy metabolism, and here should be somewhere the pathways that we will consider. So here is Krebs cycle. It is designated here as citrate cycle or TCA, and I guess the thing should be glycolysis. So maybe we will find even a glucose somewhere here, but I'm not sure how to look for that. So good. Now to the mechanism. So I just want to go through a few of the mechanisms. If we will not have many time, I will start skipping them. So that that depends. But let's get through few of the examples, and that will make you a little bit more familiar with all that push arrow mechanisms, because as I see from the homeworks practice, you probably need a little bit more practice with that. So here is the mechanisms of the human hexokinase 1. So here you see the ATP, so that's this thing is ATP. So here is OH group of the glucose. So these R basically designates all the rest of the glucose. And we are performing now a reaction to transfer this group to the glucose instead of this sort of hydrogen. So what do? We have asparagine. It abstracts hydrogen atom, so you see here O minus. It basically uses its electrons to create a new bond to these hydrogens, so a new bond is created. Now, it's a little bit bad drawn here, but this arrow should start here on this sort of bond. So it should show you that this bond, it contains 2 electrons. These 2 electrons now are used to create a bond between phosphorus and oxygen. So here will be a bond created. This arrow basically shows these electrons transferred to here. And now this bond is broken and these electrons are transferred to the oxygen. Basically, it acquires some negative charge and this bond is broken. So what do we see here? Down here after this step performed, here you see our glucose 6-phosphate. So you see phosphate group attached to the oxygen. This is ADP, so ADP is left. So you see this oxygen acquired negative charge. So electrons move to that oxygen, it acquired negative charge, and it's ready to go. This thing is ready to go. The only thing is left. We should regenerate our initial state of the enzyme. So how do we do that? We have plenty of water all over the place, and this water can abstract this hydrogen. So here we see this hydrogen abstracted, so this water is basically transformed into H3O, and electrons from this bond, once again it's not really well drawn, so it should be drawn from the bond to the oxygen. So these electrons, they fully transfer to the oxygen, it acquires minus charge, and we basically restore this sort of state that we have at the beginning. The clothing of the cleft that I showed you, that change in conformation of the hexokinase is basically to keep water molecules away when we are transferring the phosphate group. If the hexokinase were rigid, a molecule of H2O may occupy the binding site, and glucose, the binding site that we have for the glucose, because we are basically using just this OH, and the phosphoryl group of ATP will be just detached, and rigid kinase will just work as an ATPase. but it will not produce anything useful for us. It will just destroy ATP, so we definitely don't want that, and that's why we have that sort of conformation change. So it's tailored in such a way that it closes, all water molecules go away, they cannot get into that cleft, then the reaction is performed, and then it opens and everything goes away and water comes in and performs this sort of restoration of the oxygen. Good. Second step, glucose 6-phosphate to fructose 6-phosphate. You see the delta G is a little bit positive. Basically, if you see delta G, so difference in Gibbs free energy, well, from -3 to +3, you basically can count that as 0. So it means that everything is in some sort of equilibrium and reaction goes one way, another way, so as it pleases. Here is basically the reaction shown. So you see what happens. Our enzyme takes this carbon atom and basically takes it out of the ring, making this ring a little bit smaller, so it reattaches oxygen to this carbon atom. This is a preparation step for the next step that we will have that has a little bit more meaning. Once again, I have a 3D view for you. Here is one more link that I have for almost all the enzymes that leads to this sort of resource with the reaction mechanisms. So you can use this resource and just step through the steps of the mechanisms and look how all these bonds are destroyed, created, so on and so forth. I tried to copy some parts of these reaction mechanisms, but you can go there and check that resource as well. So what else should I say? Isomerase is an enzyme that catalyzes the conversion of the molecule into one of its isomers. So here the aldose glucose 6-phosphate is converted into ketose, fructose 6-phosphate. So you remember sugars, we have a little bit different sugars, and if you draw them in a linear form, some of them will have an aldehyde group, some other will have a ketone group. So ones we call aldoses, and the others we called ketoses. So it transforms aldose into ketose, fructose 6-phosphate. This conversion is essential to allow an eventual split of sugar. So I will show you that with the next steps. The delta G is about 0, so if it's less than +3 kilojoule per mole, you can consider that as almost 0. And here is a proposed mechanism of how everything is performed. So you will often see these sort of histidines. They have, you see, this nice ring, and nitrogen in this ring has two non-bonding electron pair, and this non-bonding electron pair is often used to perform something interesting. In this example, it abstracts hydrogen atom from this sort of glucose 6-phosphate. It is abstracted. Now a double bond is created, and you see how this molecule is basically transformed into a form without a ring, so it's sort of linear form. Now we have the second step. When these electrons from this hydrogen atom move here, because we are abstracting the hydrogen atom with the help of the glutamine. So here you see O minus from the glutamine, it's abstracting this hydrogen atom, and electrons that were keeping this hydrogen atom move here, creating a double bond, and electrons from these carbon atoms that were attached to oxygen, so one of the bonds is broken, and electrons are transferred to the oxygen, giving him negative charge. So you see that in this part. So here you have negative oxygen and all the rest. And now this oxygen can abstract hydrogen from the other nearby oxygen. These electrons move here, creating double bond now here. So here will be double bond. Now these double bond should be broken, and these electrons are used to attach this hydrogen, and the hydrogen leaves its electrons to this oxygen. So now we have this sort of structure. And the last part is basically we are attacking with these electrons this bond, and this bond is used to abstract the hydrogen that the histidine took from us at the step number 1. So here we had histidine without hydrogen. Now we are taking our hydrogen back. And here's basically the final result. I know that may be a little bit complicated, but just believe me, if you look for few such reactions, you will get a solid grasp of what is happening and how all these electrons are transferred here and there. Now reaction number 3, much more interesting, performed with the phosphofructokinase. This is a step that needs ATP. Once again, this is a highly regulated step. I guess we will get to that. So really highly regulated step. So I'll show you what are the regulators. But here's the interesting thing. So we had one phosphate group. Now we are attaching one more phosphate group. So we get 2 phosphate groups. So here's one more nice representation of this sort of enzyme. And here is a tricky question for you. How do you think, why do we need adding the second phosphate group? So what has so drastically changed with these 2 groups? How they may be, let's say, helpful for us? I'd even give you a hint. So at the next step, we will need to make from one molecule, we will need to make 2. So we are splitting a 6 carbon atoms molecules into 2 molecules that will contain 3 carbon atoms. So how might these sort of phosphate groups be helpful? Let's think about the charge. So what do you see? So negative charge, yeah, and we have 2 parts of the molecule with negative charges. So like charges, they do what? They repel. Yeah, so it has to do with energy. So you see what's happening. This part is negatively charged. This part is negatively charged as well. Well, it will help to split the molecule in 2 halves, because these parts with negative charges, they are repelling each other. So they are basically creating a strain on these bonds. So they are trying to pull the molecule apart. Of course, they cannot do that, so it's not enough energy to break all these bonds, but they are basically helping, because now we need to use much less energy to break these bonds. They are already strained. So there is already a Coulomb force that is trying to tear this molecule apart. Well, maybe it's too grammatic, but you get the idea. So placing 2 negative charges one near the other, we are creating a strain in bonds that will be helpful when we get basically to transforming this molecule into 2 parts. Okay, I guess we are getting a bit scarce on time, so probably I will just miss all the details of these reactions. Just few points that in the glycolytic pathway, PFK1 is a rate limiting enzyme. So what does it mean to be rate limiting? Who can tell me?
Inhibitor,
I'm sorry.
inhibitor enzyme.
Inhibitor enzyme. Well, let's put it a bit another way. So you have a chain of reactions, and each reaction goes with its own speed. So if some reaction is called a rate limiting step, what does it mean with respect to the speed of this reaction?
So this is the longest reaction.
Yeah, so it's the slowest one of the reactions that we have. Yes, absolutely correct. It's slowest step, or really you can call that a bottleneck step. So everything is okay, but this step is the slowest. So and basically the slowest step determines the overall rate of the chain of the reactions. So you need to wait until everything passes through that slowest step, and then you will be able to complete the whole chain. So this is the rate limiting step. It only works in the forward direction. So we had many other enzymes. They work one direction, another direction. This one is the committed step. Of course, it uses ATP, and PFK1 enzyme exists as a tetramer. It's like a hemoglobin, and it can exist in two states, like tense and relaxed states. So it has some sort of allosteric regulation, and it has many allosteric regulators. One important allosteric activator of eukaryotic but not prokaryotic PFK is the fructose 2,6-bisphosphate. It's formed by a separate phosphofructokinase PFK2. So this is PFK1, and there is some more enzymes. One of them is called PFK2, and it creates basically fructose 2,6-bisphosphate that regulates activity of PFK1. Good. Here are some more nice pictures how everything looks like. In this picture only a dimer is shown. Basically it should be tetrameric, but that will clutter everything too much. Once again, I give you some links where to look like and to, I don't know, rotate the structure and look in more details. Good. Next step, fructose 1,6-bisphosphate to dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. Here you see some sort of enormous delta G. It's performed with aldolates, and this reaction has an interesting twist. So you might guess that with such a large delta G naught basically, this reaction should not go in the forward direction, but it goes. How do you think why? Can you guess? What should we take into account except for delta G naught? We don't use any energy here, but reaction goes in the forward step in the cell, despite delta G naught being, well, quite high. 24 kilojoule per mole, that's definitely not negligible. Plus 3 is negligible, 24 is basically not. Pressure, temperature. No. Entropy is in some sense taken into account by free energy, because free energy contains enthalpy and entropy, so there is some sort of entropic part and enthalpic part. The twist is that we decide whether reaction is more or less spontaneous or not, or what happens to the equilibrium, not based on delta G naught, because delta G naught is for standard conditions. So yes, you have standard conditions, you have fixed temperature, you have fixed pressure, and you have fixed concentration. So your concentrations should be 1 molar, to use delta G naught as a reference. What of these three parameters, temperature, pressure, and concentration, we can change in, let's say, living things? Concentration, yes, absolutely correct. It's hard to change pressure in some biological systems. It's really not very good to change temperature, and you can do that in, let's say, in some large range, but you can change concentration. And the thing is that the product of these reactions are readily used in the next steps, so their concentration is really low. So by the Le Chatelier principle, reaction is still going into this direction, and that's because if you take into account not only delta G naught, but plus that summand that we had that contained concentration, so you remember that logarithm of Q and so on and so forth. So if you take that into account, you will see that reaction can proceed in this direction and give you the products, just because you are very quickly using up everything that is basically synthesized. And this is that cleavage step that we wanted to have to create, let's say, two molecules out of one. Here is a small table. There are a few different classes of aldolases. I'm not getting into detail, so here you see some sort of characteristics like mechanism of action, something about structure, so whatever. Here you have this nice view of the molecule, and once again some links that you can use, and here is sort of mechanism, so how it's going. We have class 1 aldolase. That's the enzyme. Well, here you see the mechanism of action. It proceeds through a Schiff base intermediate. So here you see this NH double bond, so this is basically Schiff base. Here is a Bronsted-Lowry base that will abstract this proton. So I don't think we need to get into these details, but as you see, the mechanism is more or less known. Basically, these all are tentative mechanisms, so maybe in the future something will change, maybe we will acquire some more knowledge about all these enzymes, but the best I could get for you, so it's presented in these slides. So here is an isomerase that transforms dihydroacetone phosphate DHAP into glyceraldehyde 3-phosphate G3P. And here is this enzyme. How do you think, why do we need this sort of isomerization? Any ideas? Okay, let me show you the big picture that we had at the beginning. Here it is. So this is the step we are on now. How do you think, why do we need that? if we did not have this fifth step, so and couldn't interconvert these molecules, what should we do, or what should the cells do in this case? How should the evolution go to give us something working? If we just throw away one of the molecules, that's really bad. Well, it's basically what it's doing right now. So it transforms two molecules into, well, it transforms one of the molecules into the another one to get two identical molecules. The idea is that if you do not perform this sort of transformation, you have just basically two options. So one option is just throw away one of the molecules, and that will not work because we have invested 2 ATPs, and what can we get is just 2 ATPs. Maybe some subsequent steps may get us something more, but it starts looking no good. The other option is to build this sort of pathway, but for the different molecule. So we will need one more pathway for this molecule to go through and to give us few more ATPs. So basically the idea of this step is to avoid the necessity of building one more long chain of the reaction, creating a lot of other different proteins and so on, because creating new proteins, that takes some energy, that takes some resources, and you don't want that. So you want to reuse everything that is reusable. So that's the idea of this step. So we are avoiding creating one more branch of the glycolysis, and we are just reusing whatever is already working for us. Good. What else? So it's a simple isomerization reaction, one product, it continues, delta G naught is close to 0, so nothing special here. Now, reaction number 6. So I guess we are now in the payoff step. So we start with glyceraldehyde 3-phosphate. We have basically two molecules of these, but we will consider what happens to the one of them. Of course, the same will happen to the another. Here is glyceraldehyde 3-phosphate dehydrogenase, a really nice protein, and once again you can use 3D view and mechanism to get into details. Here are details of the mechanism, pretty simple as you see. Here are cysteine and histidine residues that are used. So cysteine is basically donating a hydrogen to the glyceraldehyde 3-phosphate. Here you see NAD plus that will be transformed into the NADH. So this is sort of electron carrier molecule, and we will see that when we get to the electron transport chain. So the reaction proceeds through a Sire-ester intermediate, which allows the oxidation of glyceraldehyde to be coupled to the 3-phosphorylation of 3-phosphoglycerate. And here you see the steps. I will not get through all of them. You get the idea. Good. Reaction number 7, phosphoglycerate kinase. It's now a payoff step once again. So we have ADP and we create ATP. So we are using one of the phosphates here to be transferred to ADP and create ATP, and we are left just with one phosphate. So we get to 3-phosphoglycerate or 3-GP. Once again, in mechanism, how it's going, really simple, as you might see. So just this oxygen is using non-bonding electron pair to create bond here, and this bond is broken, electrons transfer to this oxygen, and it acquires some negative charge. So an ATP has been made for each of the two 1,3-DPG molecules derived from glucose, and we made back the ATP used in steps 1 and 3. A mixed phosphor anhydride bond is broken in 1,3-BPG as a phosphoanhydride bond is made in ATP. The mixed phosphoanhydride has higher energy than its hydrolysis product compared to the phosphoanhydride in the ATP. The reaction proceeds with negative delta G naught. Reaction 8. You see it's a little bit positive, delta G naught, but once again, everything up to 3 kilojoule per mole, that's nothing you can approximate with 0. This one, you might guess that if you keep the concentration of your product low, it will still go forward. We had 24 and reaction went forward just on the expense of low product concentration. Here's once again how our protein looks like, so how our enzyme looks like, and it's called phosphoglyceride mutase. What it does, it basically takes these sort of phosphate groups and moves it to another carbon atom and effectively swaps places with the OH group. So here you see the result. So OH and phosphate group are swapped places. So it proceeds with a little thermodynamic barrier. Its function is to locate the phosphate on the carbon 2, which on the next reaction, dehydration, will form a molecule whose phosphoryl transfer potential is greater than ATP. So we will see that. It seems so simple, but the enzymes that catalyze the reaction are diverse and quite complicated from a mechanistic perspective. So let's move. Here is the example. There are basically two different phosphoglyceride mutases, and one of them is called cofactor dependent, one is cofactor independent. We have cofactor dependent. It has much simpler mechanism. If you look with that links that I have left for you, you will see that cofactor independent is much more complicated. And where it needs the cofactor, and cofactor is 2,3-BPG, bisphosphoglycerate, this bisphosphoglycerate is used to put a phosphate group on the nitrogen atom of the histidine. Now this phosphate group is basically abstracted with 3-phosphoglycerate. So this bond is broken, electrons are used to connect to this phosphorus, and these electrons are going back to the nitrogen. So you see nitrogen has plus charge, so it used non-bonding electron pair to keep this phosphate group. Now it's broken, and you see that we got 2,3 bisphosphoglycerate, so it has 2 phosphate groups, but this is a mutase, so you should have 1 phosphate group. You just are swapping things around. And here you see that this nitrogen takes back phosphate group, but this time this is another phosphate group located on the different carbon atom. And this carbon atom takes back hydrogen that was basically donated with on this step. So that's basically what's happening. And at the end of the day, you get these sort of results. So you have 2-phosphoglycerate. When you started with 3-phosphoglycerate, so your phosphate group has moved to another carbon atom, and the state of the enzyme was perfectly restored. So pretty simple mechanism, so that's what we have. Reaction number 9, 2-phosphoglycerate transforms into phosphoenol pyruvate. I guess we saw this molecule at the beginning of the lecture, so it's highly energetic molecule, and the reaction is performed with the enolase, so it removes water and transforms that into enol. So you see this double bond here, yeah, so you remember, so enols, they should have somewhere a double bond between carbon atoms, so here it is. So now you see why did we have the previous reaction. So with a simple dehydration reaction, a molecule with high phosphoryl transfer potential has been produced, which is the next and the final step of glycolysis. Let's take a look. So here is basically how the enolase works. Once again, pretty simple mechanism, at least as it looks like. You use nitrogen to abstract hydrogen atom. These electrons are going here, creating double bond. These electrons go to the oxygen atom, giving a negative charge. Now, these, or well, actually, any of these two oxygens, they are symmetric. So either the upper or this lower oxygen, whatever, it transfers electrons back, recreating the double bond. This double bond basically shifts to here, and this bond is broken, and that's what you have, phosphoenol pyruvate. So this is the result. Good. Step number 10. We have phosphoenol pyruvate and ADP, and we get pyruvate and ATP. So one more ATP can be generated. You see this delta G naught is pretty low, minus 31 kilojoule per mole. So this reaction readily goes. Here you see the ketoenol tautomerization. I'm not sure if I told you about that in the first part of the organic chemistry, but anyway, here is an example of how the molecule so-called performs so-called tautomerization. So you get these electrons transferred to here, creating a a double bond between carbon atom and oxygen, while basically this hydrogen atom is transferred to these carbon atoms. So it's not push arrow what I'm drawing now, but you get the idea. So transferring a proton from here to here. Good. What else? I'm skipping the mechanism. Clinical significance of pyruvate. So pyruvate carboxylase deficiency. It is seen as an inborn error of metabolism where mental retardation is manifested. So it's really, really bad. Incidence 1 into 25,000 births. So it's not so rare as we wish. In these cases, lactic acidosis is noticed. Maybe we will see how that happens. Hyperthermia. This may occur when halothane is given as an anesthetic to certain persons. The ryanodine receptor, a calcium release channel, is defective. It leads to inappropriate release of calcium from sarcoplasmic reticulum. These result in uncontrolled heat generation and damage muscle cells. ATP is depleted, lactic acidosis, rhabdomyolysis, well, no good. 1 in 50,000 of population. And ethanol inhibits gluconeogenesis. I guess we will get to gluconeogenesis, I hope. And during the metabolism of ethanol, the level of cytoplasmic NADH is raised. So there are once more adverse bad effects, let's say. Here is the video for don't have time to watch that all the 5 minutes, but it basically considers the of the so if you have some time,
energetic
maybe you
glucose molecule.
Maybe you will look that when you have some more time. Okay, what do we have? A few more minutes. Good. Regulation. I told you that glycolysis is highly regulated pathway. So regulatory enzymes or key enzymes of glycolysis are glucokinase, step 1, phosphofructokinase, step 3, and pyruvate kinase, step 9. So here is basically the pathway and how it is regulated. Where you see plus means the upregulation, so the enzyme starts working harder. Where you see minus is basically some sort of downregulation, so it's an inhibition of the enzyme. Here you see examples who is activating and who is inhibiting. Probably known to you insulin and its counterpart glucagon. So one is released with that beta islets of the Langerhans, and it signifies that you have eaten something. The other is released when you have not eaten for a long time. So one tells your body that there will be a lot of glucose and some uptake should be started. So it basically activates the pathway of the glycolysis. So all these pluses, they tell that we have a lot of glucose, we had a meal just a few minutes ago, so we should start processing that glucose, storing that, whatever. The other tell us that we had no meal for some time, and probably in some places we should make this glycolysis slower. We should not use so much of the glucose and try to rely on any different sources of energy that we may have. Let's say beta oxidation of fatty acids or something like that. In some cases, for example in liver, we can start so-called gluconeogenesis. So it's like regenerating the glucose from what we have, and basically returning that glucose into the bloodstream. That is made to basically it's made because the brain needs a lot of glucose. It is unable to use many different things. It can use some ketone bodies, but that's not really good for the brain. So we are returning that glucose to the bloodstream, so we can function for some more time. And here are, of course, all the examples of what is happening and when glycolysis is favored and when gluconeogenesis is favored. Here are some factors regulating glycolysis. I'm not going through all of them, so you can read them when you have some time. Of course, all these enzymes, they are pretty important, and if you have deficiency in any of them, that's probably some bad disease. So here are some main clinical features and mechanisms when something is broken in the glycolysis pathway. Here is just once more an overview of the glycolysis, so all the 10 reactions that we had there in our scheme, and here are some here's some information on the delta G naught for different reactions. You may notice these prime here. That's basically all the conditions that we had for G naught plus pH should be 7. So if you see naught and this prime, it means that you have that pressure, temperature, so on and so forth, concentration, and additional condition that pH should be 7. And here you have delta G that is basically in the living organism if you take into account all the exact concentrations that you have in the living cell. So you see the numbers are pretty different. So here you have aldolase plus 23 and 8, so almost 24 kilojoule per mole. But if you take concentrations into account, you get minus 1.3. So it becomes spontaneous from non-spontaneous. Good. Other sugars can enter glycolysis, but they should do that through their own, let's say, pathways. So here you have a path for a fructose. So fructose metabolism is unregulated, and by contrast, glucose breakdown in glycolysis is highly regulated. And here you see how that enters the glycolysis, so through fructokinase that transforms that into fructose 1-phosphate, then aldolase B, and so on and so forth. Here you see how galactose and mannose are entering the glycolysis, so this is the glycolysis pathway and how they basically enter and what they do, so not getting into that. I know, I guess we are I have used up my time, but I'll use just a few more minutes to get through gluconeogenesis, and we will stop by that. So we have glycolysis that basically uses glucose to produce some energy, but I told you that in some cases we need to regenerate glucose in one tissue, for example, to be used in another tissue as a fuel. So it's in opposing pathway. Some reactions of the glycolysis are reversible, so it's not a problem to get a reverse direction reaction because it's reversible already. Some reactions, for some reactions, you need some bypass steps, and if you have a committed step like from fructose 6-phosphate to fructose 1,6-bisphosphate, you need a different enzyme that will reverse this step. And here is the scheme that shows you how it's all going. So here you see one step that is performed by different enzyme, and here is another step performed by different enzyme, and down here is one more step. So there are some steps that you need different enzymes to reverse. Gluconeogenesis, only liver can replenish blood glucose through gluconeogenesis because glucose 6-phosphate phosphatase is present mainly in liver, kidney and intestinal mucosa to a lesser extent, and it's totally absent in muscle. So liver plays the major role in maintaining the blood glucose level. So it basically maintains that level. During starvation, gluconeogenesis maintains the blood glucose level. So if that level falls down too drastically, we'll basically go unconscious almost immediately. The stored glycogen, so liver stores glucose in form of glycogen. Well, there are some different forms. You can synthesize fat, and so on, but glycogen as well. It will be depleted within the first 12 to 18 hours of fasting. So on prolonged starvation, the gluconeogenesis is speeded up, and protein catabolism provides the substrates, namely glucogenic amino acids. And energy requirement: the reaction catalyzed by pyruvate carboxylase, phosphoenol pyruvate carboxykinase, and phosphoglycerate kinase require 1 ATP each. So 3 ATPs are used by one pyruvate residue to produce 1/2 molecule of the glucose. So we should use 6 ATPs to regenerate one glucose molecule. That's really energy consuming. Here are some substrates for gluconeogenesis. So we can use lactate, we can use gluconeogenic amino acids, and glycerol or propionyl CoA. And important even chain fatty acids cannot be converted to glucose. So whatever went into fat, it's gone. You cannot convert that back to the glucose and use it. So that's the case. Here is an example of so-called Cori cycle, and you see that we have glucose in the liver, in blood, and in muscle. So in muscle we use glucose, we generate pyruvate. From pyruvate, we'll get to that tomorrow, we will generate lactate. This lactate can be returned through the bloodstream to the liver and then regenerated to the glucose. So muscles use glucose, they have no time to reprocess that somehow, so they return to the bloodstream the lactate, and basically the muscle pain is due to that lactic acid that is generated. Liver transforms that back to the glucose, returns to the bloodstream, and they can once again uptake the glucose and use that to perform mechanic work. So that's what we need. One more cycle through the alanine. So glucose alanine cycle, it's one more important cycle, and that's used as well. And it's as you see, it's related to the lactate cycle. So the lactate cycle is called Cori cycle, and alanine cycle, I don't know, it's called, I guess, alanine cycle. I'm not sure, why can't I oh oh yes, we are almost there, so 2 more slides. Okay, just 2 more slides and we are done. I just need to remember what that. malate aspartate shuttle. We will have some next steps performed in the mitochondria. I guess I will tell you more about that next time. But you see that different molecules, some of them can go on their own, some of them need to be transferred through specific channels, and some of them are even converted to different molecules to be transferred and then converted back. So here is malate aspartate shuttle shown, and I don't know. And here is, I guess, the last slide. It's once more about the hormonal regulation, but this time not glycolysis but gluconeogenesis. Here you see the overall view. So this is the Krebs cycle. We will consider that next time. Here is, I guess, this is gluconeogenesis shown. You remember that parallel is the glycolysis, so they are almost the same because many of the enzymes, they work two directions. Just few of them should be reverted. And what I want you to say that hormones glucagon and glucocorticoids, they increase gluconeogenesis. So glucagon tells us that we haven't eaten and body needs some supply of the energy, and liver starts depleting its stores, transforming into glucose. Glucocorticoids induce the synthesis of hepatic aminotransferases, thereby providing substrate for gluconeogenesis, so we can use some sort of amino acids like alanine. The high glucagon insulin ratio also favors induction of synthesis of gluconeogenic enzymes, and at the same time synthesis of glycolytic enzymes are depressed. So so if we start generating glucose, of course we are repressing glycolysis, because that would be a little bit stupid. We generate glucose and at the same time we are degrading glucose, so either one way or another. So that's pretty reasonable that we are either trying to generate that or we are trying to degrade it, and insulin that inhibits the process. Good. I guess with that being said, we are done. And the next time we will get to what happens after the glycolysis. Good. So if you have any questions, comments, please fire away. I guess no comments, no questions, yeah? Everybody's tired. Good. Then have a nice evening and see you tomorrow.